Viruses are unique entities that replicate within host cells by hijacking the host's cellular machinery. The replication process is a complex multi-step procedure essential to the survival and spread of the virus. Firstly, a virus recognizes and attaches to a suitable host cell through specific proteins on its surface, which bind to receptors on the cell's membrane. This specificity determines the host range and tissue tropism of the virus, meaning which species and types of cells it can infect. After attachment, the virus penetrates the host cell. In some cases, the whole virus enters the cell, while in others, only the viral genetic material is injected. Once inside, the virus sheds its protective coat, and its genetic material, DNA or RNA, is released. The viral genome then hijacks the host cell's ribosomes, the cellular machines that produce proteins, to start the synthesis of viral proteins. These proteins include the viral structural components needed to build new virus particles and non-structural proteins that can interfere with the host cell's normal functions and immune responses. Meanwhile, the viral genetic material replicates itself either in the nucleus or in the cytoplasm of the host cell, depending on the type of virus. Viruses containing RNA as their genetic material often bring their own enzymes for this process, as host cells typically do not have the machinery to replicate RNA genomes directly. Once enough viral components have accumulated, assembly of new virus particles begins, a process that can occur at various locations within the cell, such as the nucleus, cytoplasm, or at the cell membrane. The newly formed viral particles are then released from the host cell either by lysis, where the cell breaks apart, or by budding off from the cell membrane, a process that allows the cell to continue living and produce more viruses. 1. Entry inhibitors. These drugs prevent the virus from binding to or penetrating the host cells. For example, HIV entry inhibitors block the interaction between the virus and the CD4 receptor on immune cells. 2. Nucleoside and nucleotide analogs. These compounds resemble the normal building blocks of DNA or RNA but have modifications that terminate the growing chain of viral genetic material when incorporated. This halts the replication process. Acyclovir, used against herpes viruses, is a well-known example in this class. 3. Non-nucleoside inhibitors. These drugs directly target and inhibit viral enzymes that are necessary for replication. For instance, non-nucleoside reverse transcriptase inhibitors, NNRTIs, block the reverse transcriptase enzyme in HIV, which is crucial for converting viral RNA into DNA. 4. Protease inhibitors. Another antiviral strategy includes inhibiting viral enzymes called proteases, which are essential for processing viral proteins. Blocking these enzymes prevents the maturation of new viral particles. Protease inhibitors are widely used in the treatment of HIV and hepatitis C virus (HCV) infections. 5. Interferons. These are signaling proteins made and released by host cells in response to the presence of pathogens, such as viruses. Interferons bolster the immune response by signaling other cells to increase their defenses. They are sometimes administered as medication to boost the immune response against viruses like HCV. 6. Immune modulators. These substances modify the immune response to better target and clear viral infections. They may enhance specific antiviral immune activities or reduce harmful inflammation caused by the immune response. Antiviral strategies are diverse and depend greatly on the type of virus being targeted, as well as the phase of replication it is aimed to disrupt. Ongoing research continues to develop new antiviral drugs to tackle existing and emerging viral threats effectively.